Students, welcome again in our biology program. Today we are going to complete Unit 2, in which we are going to deal with gene interaction. In this chapter we will deal with some characters or the lack of dominance, some incomplete linkage and so on. But the first chapter in which we studied Mendel's law and the complete dominance. Today we are going to complete this unit by gene interaction. Let's go and see the first question. What is meant by lack of dominance? What is meant by lack of dominance? Lack of dominance occurs when the allele of neither parent is fully expressed and the offspring's phenotype is intermediate between that of the two parents. Let's begin by another question or an example for the first question. In a cross between two plants, one of the red flowers while the other of white flowers, the first filial were all pink flowers. Explain from the hereditary basis the results of the cross between two plants of pink flowers, two plants of pink and white flower. Incomplete dominance or lack of dominance in snapdragon. Here are two flowers, one is red and the other one is white. Then both of them they crossed. The first one is R capital because it is red, the second one it is white and W capital. All the offspring at the beginning it was pink, R capital, W capital. For this pink flower, we crossed two pink flower, RW capital, which is crossed with RW capital. In this table, we found that the gametes are R capital, W capital, R capital, W capital, and this represents the male while this represents the female. Gametes. The first one R capital, so it is red. The second one R capital with W capital, it means this is pink. Here also R and R capital, W capital, it means pink. And the last one, it is WW, that's why it is white. So we obtained one red, two pink, and one white. This is what we call it the intermediate color, because, or the intermediate character, because the main character it was red and white. So the intermediate character it is the pink character. Then in the other case it is a case of lack of dominance. This one because the appearance of the pink character. Red is represented by the R, white is represented by W. Pink is represented by R, W capitals. For the parents, the R, W, both of them are capital, R, W, both of them capital. For the gametes, representing the male, which is R, capital, W capital. And this is because of the presence of the pink flower. The second one, R, W, because it is also pink. So when two pink flowers are crossed together, the first it will be red, R capital, the second pink, R capital, W capital, R capital, W capital, and these are called the genotype. And the last one, W capitals, and this represent the genotype of white. So the white it is a phenotype, while the genotype it is the letters here, W, W. So the phenotype, red, pink, and white. The red is 1, the pink 2, the white is 1. For if one pink is crossed with one white, the gametes will be red, R and W representing the pink one, while the white is represented by 2 capital letters of W, W and W. The first one, it will be pink, R capital, W capital. The second one, white, 2 W's capital. The third is pink because R with the W is RW pink. And the last one, 2 W's, it represents the white. So here it is white to pink, 50-50 both of them. So if we there is 2 characters are crossed, the 2 first plant are pink, so the first generation it will be red, pink, white for this example. 2 plants of pink flowers when they are crossed together, because pink it is the intermediate character, so this will be as 1 red, 2 pink, 2 white and white. In the second example, which is 1 pink to 1 crossed with white flower, we found that the ratio is 50-50 or 1 to 1. And the ratio 1 to 1. Let's move to another question. Compare between complete and lack of dominance. For the complete dominance, dominant character appears in the first filial. Dominant character appears in the first filial. While in the lack of dominance, the intermediate character appears in the first filial. In the second filial, both characters appears, or the second generation, the both characters appears in complete dominance. In incomplete dominance, in second filial or the second generation, the 3 characters appear as the example of 1 red, 2 pink, and the third one is white. So the 3 characters appears. The dominant character has 2 genotypes, pure or hybrid. This is incomplete dominance. In lack of dominance, it is easy to identify the genotype from the phenotype because it appears as it is. So it is the hybrid one, it is the intermediate character. Example for the complete dominance: flower color in P, Andalusian fowl, flower color in Antherinum plant. The Andalusian fowl, and this is a type of birds. What will happen if white Andalusian fowl is crossed with a black female? Explain the offspring of the first and second generation. Here an example. This is the white fowl and this is the black one. When they are crossed together, the first white is WW capital, while the black BB capital. When they are crossed, all the offspring is blue or intermediate character in between. So B and W capitals for the first generation. In the second generation, if two blue are crossed together, we found that the second generation there is a black, which is 1, and this is B capitals, while the two blue, each one has the genotype B capital, W capital, and the last one, it is the white, which has the WW capitals, both of them. This is a non-Mendelian case. White W, black B. The blue B capital, W capital. For the first generation, when the two vowels are WW, which represent the white, while B capital, which represent the black one, when they are crossed together, the gametes are W capital and B capital. When they are crossed together or combined together, they form WB capitals, W capital, B capital, and this is the first generation, which is a blue one. So 100% of this offspring is blue. And here's the blue fowl. For the second generation, when two blue fowls are crossed together, B capital, W capital, B capital, W capital. This is the blue fowl. When they are crossed together, the gametes BW for the first one, BW for the second one. The second generation, or filial 2, B capital, B capital. This is a black. W capital, B capital. This is blue. B capital, W capital. B capital, W capital, another blue. And the last one is WW capitals, and these are white. Here's the black, blue, white. So one black, one white, and two blue. One black, two blue, one white. And now, dear students, we have studied one of the non-Mendelian cases which represent the lack of dominance, in which we knew that there is two dominant characters. When they are crossed together, the intermediate characters appear. And now, what about the other characters? Is there any other non-Mendelian case or not? Let's go and see what will happen after that in our session. What is meant by complementary gene? Give examples. What is meant by complementary gene? Give example. It is a case that is controlled by two pairs of genes. It is a case that is controlled by two pairs of genes. Second, the dominant character appear only if the two dominant different genes are present in pure or hybrid state, where each one complete the function of the other. Another time, it is a case that is controlled by two pairs of genes. The dominant character appear only if the two dominant different genes are present in pure or hybrid state, where each one completes the function of the other. How this takes place? Both pairs of genes participate in the color production, since each one of them controls the production of a certain enzyme, which affects part of the steps of the pigment production. The ratio of the second generation is 9 dominant to 7 recessive. Example: the inheritance of color in pea flowering plant, the inheritance of color in maize grains. An example: when two strands of white flower pea were crossed, all the flowers of the first were pink. In the second generation, the flower produced were 9 pink to 7 white. Explain your answer on a genetic basis. For the parents here, AA, BB, and each one is one pair of gene. In the white one, AA small, BB capital. For the gametes here, we found that it is A capital, B small, while here is A small, B capital. All the offspring, it was pink because it was A capital, A small, B capital, B small. So the flower here are pink. Let's go and see what happens if two pink flowers are crossed together. A, B, A capital, B capital, that represent A capital, B capital. This is the first gamete. The second gamete, A capital, B small. The third gamete, it will be A small, B capital, A small, B small. In the gametes for the females, A capital, B capital, A capital, B small, A small, B capital, A small, B small. For the first one, it will be A capital, B capital, B capital, and this represents the pink one. The second one, A capital, B small, B capital, B capital, B small here. So it is also pink. Why? Because one gene is into a hybrid case, and the second one is in a dominant, pure dominant. Here, the third one, A capital, A small, B capital, B capital. So this was a hybrid, while the other one is pure and dominant. The fourth one also the same, because it contains one hybrid, and the both of them, they are hybrid. A capital, A small, B capital, B small. So both of the two genes are hybrid. Here we found that the second gamete with the first one, it makes two dominant character. Here A capital, and here B capital, B small, and these are the two genes here, two pairs of genes are in a dominant case. One is hybrid, the other one is dominant, pure. And this makes the pink color appear. While here we found that one of the two genes are in dominant case and the second one into a recessive case. That's why it doesn't appear the pink color and the white color appears only. Here also we found that there is one hybrid dominant and the second one is recessive character. That's why the white color appear. Look here, the recessive character and dominant character. So in order that these characters, the pink characters appear, at least two of the pairs of genes, the two of them are into a hybrid case, or the character will never appear, or one character dominant and the second one is hybrid, one character is a dominant character and pure, while the other one it is into a dominant but hybrid one. For this, in order that both of them they complete the function of each other, and they make enzyme which helps into the production of the pigment's color. Explain how the validity of complementary gene was confirmed or was proved. Here a scientist makes some experiments on the flower to prove the complementary genes. This was done experimentally by mixing together the extract of the flower from both strands in the test tube. The pink color appears as a result of the presence of all the enzymes and the completion of the reaction. And now, dear students, we have studied one of the non-Mendelian cases which represent the lack of dominance, in which we knew that there is two dominant characters. When they are crossed together, the intermediate characters appear. And now, what about the other characters? Is there any other non-Mendelian case or not? Let's go and see what will happen after that in our session. What is meant by lethal gene? In some living organisms, including man, some genes, when found into homozygous state, that leads to the retardation of the growth and cause death to almost a quarter of the offspring. This, as in sickle cell anemia and other forms. What is meant by sickle cell anemia? Explain. Sickle cell anemia, it is a disease arising from genetic disorder in the gene responsible for hemoglobin synthesis in the red blood cells. Sickle cell disease involves irregular sickle-shaped red blood cells caused by abnormal hemoglobin. SS represent normal hemoglobin and SS small represent the sickle condition, usually die before maturity, while S capital, S small, or the hybrid one individuals have the intermediate condition called sickle cell traits or carrier for this disease. For example, if two carriers are crossed together or married, what will happen? The gametes for both of them, S capital, S small, S capital, S small for both of them. For the second generation, S capital will appear. The second generation for the S small, S capital appear, and this is a carrier for sickle cell disease. Another one, S capital, S small, so it is also a carrier for sickle cell disease. The last one, it will die because it contains two recessive characters, S and S small. Let's go and see what are the phenotypes. This is normal. Both of these S capital, S small are carrier, and the last one is a sick person and it will die before maturity. Give short account about multiple allele, then give an example. Multiple alleles, it means there is more than one pairs of genes which affect the character, more than one or two genes also. Two pairs of genes. Multiple allelic traits. Multiple allelic traits. Inheritance by multiple alleles occurs when more than two alternative alleles exist for a particular gene locus. Each individual inherits only two alleles for these genes. A person's blood type is an example of a trait determined by multiple alleles. The blood types as example for multiple alleles: A, B, O, and AB are the blood types. A person can have an allele for an A antigen blood types A or B antigen blood types B. Both A and B antigens blood type AB, or no antigen at all in blood type O. On the red blood cells, all of these antigens are found on the red blood cells. Let's go and see what will happen for this. Human blood type can be type A, and the genotype of this type A is A capital, or A capital O. Blood type B, the B capital or BO. Blood types AB, it will be AB only, or the types B, it will be OO only. So the blood type A, the genotype AA or AO, blood type B, BB or BO, blood type AB is AB also, blood types O is OO also. If you look here and remind you with what happens when we added the serum antigen A and antigen B, and we add both of them for droplets of the blood. So the genotype AA and AO, we found that when the antigen A is found, so we found that they are positive, or they respond by making agglutination, while in antigen B there is no agglutination. So the blood group of this, it is A group. Also in the genotype BB or BO, here for blood group B, we found that there is no agglutination with antigen A or serum A, while in B we found that there is agglutination takes place. That means this blood group is B. Also for the blood group AB, there is agglutination for both of them because of the presence of the antigen A, antigen B in this blood group. So the blood group here is AB. While in blood group O, we found that the blood group O, there is no agglutination taking place. That's because of the absence of any antigen. So the blood group O, we call it as a universal donor, because it can give its blood to any other types of blood. So a transfusion of blood can be made from O to A to B or blood group AB. While O, it can't take any blood except from O only. Also, we considered AB as a general recipient because it takes, or the universal recipient, because it takes from all the blood groups AB. Now, dear student, we had reached the final point of our session. See you next time. I hope you enjoyed it. See you, and till we meet again. Bye-bye.